I will tell you a little bit about the infections, how we approach this question. Infections are, of course, they are very important for our health from many aspects. We all know many examples: helicobacteria, papillomavirus, HIV, so on. And if we look at the immune-mediated diseases, there's also a lot of evidence that infections may play a role. I just summarize a few of them. Type 1 diabetes, there's some viruses which are suspected. Enterovirus, for example, seems to go to the pancreas, and that's one mechanism. Adenovirus, norovirus as well have been linked to type 1 diabetes. Celiac disease, we know it's caused by gluten, but gluten is not enough, so you need also something else. Enterovirus, adenovirus, parechovirus have been linked. Asthma is a classical example where rhinovirus and RS virus is known or are known to exaggerate the disease and trigger the asthma exacerbations. And atopic sensitization, we know the old hygiene hypothesis, which actually shows that the older sibling presence of the older sibling in the family decrease the risk of atopic diseases in the newborn. So it fits very well with the virus infections because the older siblings bring the viruses to the family from daycare and school. And then if we look at the other immune-mediated disease, there's a lot of evidence. I just took one of them, MS disease and the Epstein-Barr virus, which has been a hot research topic just recently. What we do in HEDIMED here, so we try to fill the knowledge gaps. So we don't aim to necessarily confirm associations which have been already found in previous studies, but we especially want to address the weak points where you need more knowledge. To do this, we use different kind of strategies. One is to use exploratory approach. So rather than focusing on candidate microbes or viruses or microbiome patterns, we want to look at the holistic whole picture. So this means we use modern sequencing technologies to analyze the whole microbiome and virome. We also want to know more about the metabolic activity of the microbiome. And we measure the metabolites, for example, from the stools. And we use a large arrays which can find a wide range of different microbial antibodies. And we will hear a lot more about that in the next talks. And food microbiome is also one interesting. So it means that what we eat and how the for example, apples are providing microbes to our gut system. And the environmental microbiome, of course, which Aki already presented. The other thing we do is to study the host response, how we react to the infection. We also especially look at the prenatal time, already in utero infections and the early postnatal infections. And we use the intervention trials to help the evaluation of causality of the associations. And I think this is also very important advantage. We, for example, use these biodiversity exposure trials, which Aki just described. We also have a probiotic trial going on in celiac disease. This data can help tremendously to understand what factors could be causally associated with the diseases. And to further evaluate the causality, we aim to look at the mechanisms, especially using organoid models and cell models and enterovirus infection in those models, which link to T1D, diabetes, and celiac disease and viruses. So this is like one of the, I think, areas where the research is really expanding now. So there already is some information, but maybe not so much as in diabetes and asthma. So this is the previous or the state of art where we started. So there's evidence that some viruses are linked to the celiac disease, and possibly because these viruses seem to be enteroviruses, which replicate in the intestinal mucosa. This makes it possible that they can somehow trigger the immune response to gluten because they cause inflammation. And inflammation is usually a warning sign that to the immune system that something dangerous came into your body, and the immune system starts to attack the gluten, for example. And how we have done this, again, this is a good example of prospective design. We have the prospective birth cohort here, different age categories. We have a case child who was followed from birth and developed celiac disease, and 2 control children. We measured virus antibodies and virus RNA from these time points from stools and from serum over time during the follow-up of the children. and we compared the case to control children. And we found some evidence, and then also some viruses we couldn't find any association. So the viruses we found, a risk association was enterovirus and parechovirus, and these are from the DIP cohort, the Finnish cohort. And the parechovirus was previously reported in a Norwegian study as well. So this in a way confirms that. This is still unpublished finding. And also the virus detection from the stool. The previous slide was antibody results, and this is direct virus detection from stools. It also found the parechovirus. Enterovirus was not significantly associated, but it was showing the same risk trend. Then if we look at the antibody arrays in T1D process, we could confirm that enterovirus antibody responses were more frequent. For example, here the POLDID ones show significant association with the development of diabetes in the prospective birth cohort study. Again, the DIP study here. In this example, when we look at all other viruses we had in the array, we also found some trends for Helicobacter, for example, and for Yersinia. The Yersinia was almost significant after the correction for multiple comparisons. The others were not. Again, one other kind of example is the stool multiomics integration we do in type 1 diabetes. So we try to understand in a new way how the microbiome can influence our immune system. So this is a little bit complex figure, but I would like to pay your attention to a few things. So first we analyze the stool microbiome. We classify them to the microbial species, and we found some which link to the autoimmune process, but we also do metabolomics from plasma and from stool, and we do also volatilomics from stool. So volatilomics means the volatile compound, and we use two kind of technologies. The olfactomics is it's a system. It's like an electronic nose, a portable device which can detect the volatile compounds, and this is the gas chromatography method which do the same. And then stool metatranscriptomics, and we combine all these things together, and of course the outcome, which is type 1 diabetes in this case. And it seems that we can find really interesting findings already. Beta-in, for example, high in controls, and those kind of correlations between microbes and the metaboloids seems to pop out. Not published yet, so if you are interested, follow the webpage. We will inform the progress there. My last example is the in utero exposures, and as I mentioned earlier, this is again important knowledge gap. It's hard to study because lack of the good prospective materials. In HEDIMED, we have these unique birth cohorts and samples taken during pregnancy and cord blood samples, and we decided to explore in detail the inflammation during fetal life and also correlating that to infections. So we measured more than 90 different inflammation-related proteins using Olink technology and antibodies against 40 different microbes from cord blood samples and pregnancy serum samples, and associated with them with the different immune-mediated diseases in the offspring later on. And basically this was done in 4 birth cohorts, COPSAC, PREDICT, GDME, and DIP study. We had these samples from different time points in pregnancy, first trimester, second trimester, or cord blood, and then we carried out the Olink protein analysis and the array-in-well virus antibody analysis. And some of the patterns are listed here. We found we analyzed them in different levels. So unsupervised patterns, there was some patterns with linked to T1D and also in atopic dermatitis. When we went further to the single protein level, we found, for example, tweak protein, which was maybe the strongest finding in this study, and it linked to T1D in especially one of the birth cohorts we had, and some other proteins as well. And the tweak was also confirmed by multivariate analysis and some of the others as well. Again, this is still unpublished, so we try to correlate this now to the virus antibodies and microbial antibodies, and that work is still going on. As a conclusion, I would may be mentioned here. Of course, this is just a part of the studies we are doing, but maybe gives you an idea that what's coming out. So, for example, we found the association between enterovirus and parvoviruses with celiac disease. We found evidence that the neutral period may really already be important in these diseases. And the new technologies seem to be very powerful generators of new knowledge. I encourage you to follow our website to see how things progress. This is all from me now. Thank you.